Perfect, Andi.
Is everyone clear about this? Okay,
Yep.
good. So the metal is a bit different from the fire, but I think the content is the same. So I'm just going to the second slide then. Basically, I'm going to share 4 different items. One is about the dose mortality relationship, and then the second one is going to be selecting the bioassay procedure, because this is going to be very important for everyone who are going to do some bioassays. And then basic principles in the execution. And last one, I'm going to close with data analysis and usages. And I'm going to spend just a bit of time for the last item, because my colleague Alan is going to be presenting following my presentation on this issue. So I think she will be having more time to discuss in detail. But start from the beginning. I'm going to say that when we do the bioassay, I think there are 2 aspects that we have to take into account. One is the statistical aspects, and the second one is the biological meaning. And those are very important for us to have the data meaningful, while still also compliance with the statistical requirement. So for anyone who's studying about the toxicology, this is the basic understanding in regard with the insecticide, not only the chemical synthetic insecticide, but also some of the entomopathogenic material also have the same principle. Basically, this is what is called the biphasic effects, or in some literature, it's called the harmless effects. But if you look at on the blue line here, I put the line here just to differentiate between the left and the right side. And the left side means that the green line means the stimulation. So that means that if we apply insecticide and then the insect are still alive, so the effect that we may get is actually stimulation, not mortality, but stimulation. However, if the dosage is high enough and is sufficient to kill the insects, so the effect that we will get is the right side, which is the red color here. That means it's mortality. So this is the reason why having the right dose when you apply pesticide or insecticide in particular is very, very important, because sublethal dose or sublethal concentration may lead to the stimulation of the insect to actually survive better, or could be having more offspring than if they do not get any exposure to insecticide. So having the right dose, right concentration is key. Okay, there are several different parameters to measure the toxicity of the material or insecticide in this case. And it depending on the purpose of your research, or it depending also on the material that you have, you may end up having mortality as your end of the observation. But if you are working with, for example, semiochemicals or some other things that may not cause some mortality, but you can still measure the effective concentration or the inhibitive concentration. Let's say if you are doing with some enzymatic reaction. So this actually not only for the LD or LC50, but actually the principle can be used for different purposes in research. Okay, so this is very typical, what do you call, dose mortality relationship. If you look at on the x-axis, that's the doses or the concentration that you are going to use on your bioassay. And then the y-axis is actually the response, which can be mortality, can be crude inhibition, can be, let's say, if you are working with attractant, maybe how many percentage actually attract your insects or repel your insects. So you can change different parameter for the y-axis. So as you can see that the relationship is quite typical, and it's going to be hard for you or for us if we try to fit what kind of relationship having this kind of curve, which is going to be a bit difficult. So I think people then change the type of the relationship by converting or by transforming the x-axis, in this case, into log, becoming log dose or log concentration, while the y-axis is still similar. And you can see here the graph becoming sigmoid, which is going to be a little bit, if you look, it's easier and probably more familiar to us, right? So let's take a look on the three areas that I put in arrow here. This is the area one, this is the second, this is the third area. What I'm going to say here is that if we are working with insecticide or doses or concentration cause mortality only in this area one, it's going to be different result. If you are working with the same insecticide, but when you are doing bioassay, you select actually doses or concentration that fall into the category in the area 2 and also in the area 3. Because what does this sigmoid type of data means is that here in the area 1, if you increase the concentration of the insecticide or the dosage concentration with the same increment, with the same unit, actually additional mortality that you will have is small here. And it is going to be similar when you have your range of your concentration that falls in the area 3. But here in area 2, where if you increase 1 unit of concentration or dose, you actually getting more mortality or more response. So since profit analysis is actually working with the regression line, so as you can see here, in the area 2, basically your line is very much similar with a straight line. And probably you will ask, why is it 50? It's the most common number that is being used to measure the toxicity or different kind of chemicals that we want to test. Actually 50% is falls into here. And this is actually the maximum slope, or it's sometimes we call as turning point. So that's the maximum slope of the line. If we are trying to separate this line into very small fraction, there's a combination of slopes, and the slopes at the 50% is actually the highest one, or the turning point one. So that's the reason why this graph becoming sigmoid. Okay, let's say if you have area 1 and area 3, yeah, and then area 2. Here, if you do the analysis using any kind of program that Valen is going to be delivering next, you can see here where it's the ILC50. If you have the area 1, your data is falling into here. You force your data to be submitted to the profit line using any program. So what you will end up is the LC50 or LT50, which is going to be somewhere around here, because this is going to be extrapolation. It's the same thing. If your data is into this area, which is the area 3, and you are going to estimate the LC50 or LT50, the program will extrapolate into becoming this line, and your LC50 is which is going to be somewhere around here. Using the same insecticide, the same insects, the same presence, but you are selecting the right concentration of the doses that you are doing to estimate the LC50, which is the green line here. Your LC50 is going to be here. Okay, so this is something that we have to remember, because this thing will not mostly be presented in the article or in the paper that you want to write, because they will ask you probably the LC50 value. We, as a researcher, know where the LC50 coming from. Okay, and the LC50 can be different, although the insecticide and the person who are doing the bioassay can be the same, similar, can be the same. But if your behavior of the data that you are using, submitting to the profit are different, the result is going to be different. I think that's very, very one principle that you have to remember. We have to remember when we are doing the bioassay. Okay. Then the next question is that when do we need to terminate the observation after we select the bioassay procedure, or at least we know the statistical consideration, and how do we select? Is it 24 hours? Is it 2 days? Is it 4 days? Is it 7 days? It depends on the insecticide that you are testing. This is just representing you some insecticide that work on the nervous system. And usually the response of the insects that are being treated with nerve insecticide is going to be very, very quick. Only within minute or even second, we can get the result. So 24 hours is going to be probably sufficient to measure the activity of the toxicity of the insecticide. But what about if we have a slow action insecticide? For example, insect growth regulators. It takes few days to kill your larva. And some larvae may still alive even after 7 days and will continue their development. And mortality that we can get may be up to when they try to molting into the becoming a pupae. This is some of the pages that just to represent that sometimes the response or the action of the insecticide may take longer than the others. So be sure that you know what you are working with, how do you need to measure, and for the sake of the safety, I think it's going to be much better if you do not know yet your speed of action of your material. It's going to be one cycle. It's going to be much better, but then you will have a complete picture on the activity of your product. Okay. Second is how to select the bioassay protocols. Yeah, it depends on your purpose. Are you going to measure the toxicity in the term of LD50 or LC50? Because this will determine what kind of bioassay that you will be using. Not all bioassays can be used to determine LD50, or not all can be used for LC50. So you have to be sure which unit are you going to present. That will determine your bioassay protocol. Second, and then you have to think about whether are you going to use natural diet or artificial diet. Sometimes it depends on the availability of the mass healing procedure. If artificial diet is available, in many cases, it's much better because it's going to be less laborious than using the natural diet. The third one, we should also know about the mode of entry of tested insecticide. For example, if you are working with Bacillus thuringiensis, and then because we do not know the mode of entry, and then we try to use the topical application by putting some solution of PT on the surface of the insect in the human, we may end up no mortality because PT toxin will only cause some toxicity if they are ingested by the larvae. So again, understanding mode of action, mode of entry is going to be another things that we have to consider in doing bioassay. The fourth, I already mentioned, for the speed of the action. And then the fifth one is the insect life stages and feeding behavior. Which larvae are you going to test? Newly hatched larvae, third install, or probably later install. Or it could be the adults if we are working with, for example, front plant molter. So those are I think, five things that we have to think and take into consideration before we are selecting the bioassay procedure. So in principle, there are 3 or 4 different things. One is insect keeping. The second one is topical application. The fourth one is fumigation and residual assay. And then the last one, injection. So I'm just going to show you some of the pages to get things a little bit easier to be understood. This is the diet overlay. So basically, we have a diet that that are used for reading different species of Lepidopteran. So when we do the bioassay, including using the PT toxin or insecticide, we in many cases using this diet overlay. So basically, we use trays depending on the size of the trays. This is, I believe, it's a big tray. So we just put the diet into the tray, and then after the diet solidified, and then let it dry, and then we can put the insecticide solution on each well. And it depending on the size of the well, it will be determine how much solution are we going to put. So if you have different concentration of insecticide or PT toxin, and then you pour 50 microliter each well, you will end up having different doses or different concentration in each well because the concentration of the solution is different. So this is very common procedure for doing different species of Lepidopteran. We use for Asian conporer, mealworm, and then sometimes Spodoptera isigua, and also Spodoptera litura. The second bioassay procedure is diet incorporation. So if you have a lot of material, this is going to be a good choice because if you pour your insect diet into the diet and mix them up, and then when you overlay on the tray like this, and then you cut into pieces like this, so basically anywhere the larva will feed on the diet, they will ingest your insecticide. What is the difference with the overlay? The overlay, the insecticide or the protein of the Pt toxin that you are putting on that well will stay on the surface. So once the larvae get into in the middle of the diet, it could be that the larvae will no longer ingest the toxin or the insecticide. And this is going to be different with the diet incorporation. But diet incorporation need a lot of materials. So if you are only having very small amount, don't choose this procedure. Okay, the next one is dipping. Dipping can be also different purposes. You know, if you are working with, for example, diamond backmouth, Butela silostella, this is the bioassay that we usually use. So we dip the cabbage leaf, depending on the number of larvae, depending on the space that you have, you can measure the size of the leaf that you are going to dip. So this is going to be very easy because you do not have need a lot of measurement. Basically, make the concentration that you want to test, and then dip, and then air dry it, and then after that put on the cups or on the petri dish, and then release the larvae, and then do the observation. This is also another examples of dipping. So when you have an artificial diet, basically artificial diet also can be dipped into the solution. If in the case of brown panoper, we use the rice seedling, and we usually we keep the whole things, including the roots of the seedlings, and we air dry it and then put in the place that we want to use for releasing the larvae. Okay, this is another tools that we can use also for bioassay. In particular, again, when you have very small amount of insecticide, this is a good choice. It's called the water tower spray. So basically, we put the solution on the top, and then using the compressor, it will cause some hard push on the bottom, and we could put the insects or the leaves that we are going to use for feeding the on this side. So it is very powerful tools, and it produces a very small droplets. So if you want to look at the systemic activity of the insecticide, and then only one surface of the plant that you want to spray, this is going to be a good choice because you can spray on the leaf surface, but leaving the bottom of the surface will have no any exposure to insecticide, and then you can put your insect at the leaf to see if your product having some capacity to penetrate the leaf tissue so that the insect will die. So again, and this is the topical application. So to the best of my knowledge, this is the only what do you call common and practical bioassay that will allow us to calculate or to estimate the LD50, because the LD50, one thing that we have to remember is one, we have to be able to know exactly how much insecticide is actually into the insect body. Second one, we have to be able to measure the weight of the insects that we are going to test it, because the LTV is usually represented by weight per weight. So we have to know how much insecticide getting into the insect's body and how much weight per insects that you are using for the bioassay. If you are not going to be able to measure how much insecticide get into the body, you will not be able to determine the LTPT. What you can do is estimate the LCPT. Okay. So this is residual activity. We do this procedure, especially we are working with the parasitoid to look at the impact of, for example, insecticide on the adult of the parasitoid. So we usually using the insect residual activity. We use the acetone as a solution, and then the solution uh, it depends depending on the size of your tubes. If a bigger tube need more volume, if smaller tube need less amount of solution. So when you pick up and then put the solution inside your tubes, and then you just need to stir and to roll, so that all the surface of the tubes will get some solution, and then let it dry, let it acetone evaporate, and then you basically have your insecticide over your internal side of the tubes, and then you can release your insects. It can be larva, it can be parasitoid, depending on what insect that you are working with. But in many cases, we're using this approach when we work with the parasitoid. Okay. The next question is that how do we select insects? So Valen is going to be sharing with us how difficult it can be when we are in the bioassay, and but sometimes how can be it can be easy as well, you know. So having a good preparation and a good consideration is going to be a lot of things to be put into consideration. That's going to be helping us to have a good data. So make sure that we have uniform insects. Let's say one bioassay unit 500 larva larvae. So all those 500 larvae must be healthy, and then same size, same age, and different biological character that we may have. Okay, so healthy insects is going to be very helpful, because if you have insects that you are using for bioassay with different level of healthiness, variation is going to be very big, even variation within replicates, or even variation between population. Okay. The second one is that the population or the larvae that you are using also representing the population. And because, you know, the larva that hatch by today might be different from the larva that hatch tomorrow and the day after tomorrow. So if you are working with that population, then produce different eggs on different days, how are we going to select those larvae to represent the whole population? So in many cases, what we do is that we will do different replication on different days. In that case, we can have several days of assay that may be more representative to the population that we have. So try to avoid to do bioassay for 5, 6 different replication on the same day, because if you do that, I think genetically the eggs that you are using today is going to be different from the day that you are going to use for another day, because the females that produce eggs might be different genetic background than what we use today. So spreading the replication on different batch of larvae is going to be much better to have a better representation of the population. So this is just one example. This is for Asian concord. We do use jar because we have eggs here, and if the larva hatch, the eggs will be start crawling. So if we have a lot of larvae hatching on the same day, and they start crawling on the paper on the jars, so when we open the lid, all the insects, all the larvae will come up and stay on the lips of the jars. So there's a good way to know whether the larvae are healthy or not, because the larvae that are not healthy will most likely stay on the bottom of the jar. So those who are coming up and stay on the lip of the jars, they will be good for being selected as larvae that fulfill the requirement of the healthiness. So designing the reading procedure and designing the bioassay could be two different things that will help each other. Okay, because this is part of the reading procedure. Also, it's going to be useful and very helpful when we start doing the bioassay, because with this one, we can select the larvae. They are very uniform, healthy, and also what do you call it? Hatching on the same day. So once again, this is the next is about executing the bioassay. I think it's very, very important for us to optimize all the bioassay procedures before we start doing the full bioassay. What I mean is that please work with your control until you very small mortality in the control. If we refer to Apollo's formula, Apollo's formula allow us to have 20%. But in experience, 20% mortality on the control is still too high. So if you have a good bioassay procedure, it's very replicable and usually mortality ranging from 0 to 5%. Okay, so that thing have to be set up before you start working with any insecticide. Second, determine the working concentration that will give a range of mortality to 90%. This is really referring to the area 2 that I described in the beginning of my slide. So 2 to 99% is going to be a good number if we are to run the bioassay and then estimate the LD50. Carry the full bioassay with at least 7 concentration with the control. Okay, make sure that you have a plenty of insect and always use the fresh insecticide solution. So Val and I usually, before sending the data into the program, we plot the mortality data on the log paper to see the scatter of the data and see if changing is needed. So I think it's very good practice if, every time you finish your bioassay, then just plot your data into the paper and then see if the data is good or not. Because if you don't do that, and then you basically you've done your bioassay and it's just you have the data, and then later on you do start analyzing. At the end of the when you do analyzing, and then you have a very high chi-square because the variation very big between replication and between also concentration doesn't make really good sense. So have plotting on this log paper to see at the beginning of the or at the end of the observation before submitting the data to the computer programming, I think that's a good practice. Okay. I think I'm coming to my last items. Analyzing and things to watch. Against control mortality. Second, variation among the replication. Third, the pattern of the mortality data. Is it higher concentration cause higher mortality, or the other way around? I think Valen is going to be discussing on that in very more detail. And then the last one is that, are we going to use the data of 0 and 100% for the treatment? It depends, yeah. But I suggest to use 100% that cost, or the lowest concentration that costs 100% should be using in the profit analysis. But if you go back to the sigmoidal shape, you can end up having maybe 2 or 3 highest concentration of your biopsy causing mortality, 100% mortality. If this is the case for you, don't use all this concentration that cause mortality 100% in your analysis, because it's going to be causing some skew toward to the right. So, but using the lowest concentration that cause 100% mortality is going to be good for you, for us, because, and then if we want to determine LC99, and then we can check how big is the LC99 based on the profit line. Okay. And then from the practical standpoint, where is the lowest concentration that causing 100%? So LC99 of your estimate is above the lowest concentration that causing 100% of mortality. And then we can have a question, is it real? So this is a way to check your data and your result of your analysis. So this is just some of the publication that we have based on the bioassay experiments on different purposes, different issue. As our research focusing on the insecticide resistance, this under the paper that related the resistance using the procedure of bioassay that just described. Okay, so this is the suggested reading. I think there are 3 good textbooks that you may take a look and read it because it has a lot of information on it. So I think thank you very much for your attention. And this is just one thing that I also presented to you that if we work in the regional area, I think it's better for us to have a standardized bioassay protocol so that the data coming from each country or each researcher or each lab and then can be comparable because we cannot compare the data of the bioassay from the bioassay. They don't use the same bioassay protocol. So having the same protocols is going to be helping us to work in much bigger area because it's going to be comparable to any results that using the same things. So once again, thank you very much for your attention. Thanks to Alison, and I do really apologize for the delay because some issues that related with my computer. So thank you very much, and I return it to you, Alison.
Thanks, Andi. That was perfect. You don't need to apologize. You started on time, and that was a fantastic presentation. A few questions now, but we'll come back and ask some questions to everyone. But there's a few from the audience. I have a few, and Tech has a few too. So what I will do, I may ask a few of those just for time. With the larvae that you're using, is there a specific age though that is best? Because there's obviously quite a difference in larval size, like for fall armyworm, for example, or is it just any larvae?
Well, it depending on the purpose and the insecticide. Let's say you are working with the toxin. For example, you are working toxin that is being expressed in the transgenic crops. For example, Try1AP or Try1AC. I would suggest to use the newly hatched larvae. Why is that? Because basically the larvae will eating on the transgenic leaf when they hatch. So it's going to be more natural. Eh? But if you are to determine the LD50, and then you have to put 1 microliter solution on the thorax of the larvae, using first instar larvae is going to be very, very challenging. It's very, very difficult because the larvae is too small. So in that case, the third instar larvae are usually suggested. And so again, so the behavior of the insects in the nature is going to be one thing that we have to consider to select which type of larva or which size of larva that we are going to use. But in any stage of larva that we are going to use, it has to be standardized. The age of this the larvae, and also the instar of the larvae. So the bigger of the larvae is going to be more challenging because, for example, we determine use the third instar larvae. Some larvae will become when they are on the 6 days, but some may be on the 7 days, some may be on the 50s, right? In that case, and then you have to have maybe, for example, you are expecting to have 300 larvae, and then you have to start with like 1,500 larvae at the beginning to be able to have 300 larvae in the same instar, the same age, and the same size. So it's going to be more complicated to use a bigger larva.
Excellent, Andi. And I think that answers this question. Should bioassays be carried out using lab-established populations, or can it be collected freshly from the fields? And I think your answer there also answered that question. But any other insight you wish to add on that point?
Yeah, having lab population is good. We have a drone plant hopper that has been in the lab since 1986. So this is going to be very great population as a rate population because that population has no exposure to any kind of insecticide since they are in the lab since 86. So if you are trying to study about the how the feed population respond to certain insecticide, and then this population can be a reference, can be the standard. We'll be collecting from the field and then comparing the response or the toxicity of the insecticide to the laboratory in comparison to the field population. So again, both are needed depending on the population, and in many cases, if you are doing some resistance studies, sometimes it's impossible to collect insect as many as we want with the same category, uniformity, and then right away bioassay from the lab, from the field. So in many cases, we have to wait until F1 generation, or in some cases, second generation, but no more than second generation. The third is going to be different because when we are bringing insects to the lab, and then the genetic background will be different, the fitness is going to be different. Uh, the 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 more generation, the less natural they are, I guess.
Yeah.
Yeah.
And I'm quite interested about the different diets. It obviously takes quite a bit of trial and error getting all these things right. And it was really interesting to see just all that trial and error and mistakes and then improvements that you made. I think you said over a year or up to a year of kind of fine tuning
a lot
it.
of time. Andy can agree with me with that because it's really difficult.
And
Really.
do you think that's going to be the same? Is it the same for the red palm weevil? Do you think you've learned a lot from the fall armyworm, or is it every insect is different, so you have to sort of fine tune it?
I think when we are quite familiar with different species of insects, that will help for us to solve the problem even when we work with a new species, I think. But I think the basic principles that we have to understand is how the insects actually live in the nature. Because even though we can use the same diet, but the surface of diet is different. It's going to be different. For example, if you provide the diet which is very, very smooth for the first instar larvae, they won't eat it because it's too hard for the very small larvae to chew. But we provide the rough surface of the diet with the same content of the ingredient that work well. So it's not only the chemically, but also physically. It's going to be very important when we work with the diet.
Okay, so they're like kind of gourmet eaters, gourmet consumers. these pests. Andy, I see you had a comment. It just went to everyone, but I'm just going to tell I'm going to say what that is to everyone, that you've been rearing fall armyworm since 2019, since that first outbreak, I guess, was notified officially in Indonesia, and you have a recipe that you've fine-tuned. Is it similar to Davina's? I mean, it's interesting to see that some work on you sort of have slightly different maybe diets, but you also use it for the Asian corn borer species. So they seem to all like the same thing, and it must make it a bit easier. But is there a difference between the species?
Yes, if I look at the Divina's presentation, it's different. And I think first we developed the diet actually for Helicobacter pyrrhia, and that's the beginning. And then we try to play around, and the same diet can be used for Helicobacter and then for Asian corn borer. So when the fall armyworm came here, we tried and then worked for fall armyworm and also for 2 different other Lepidopterans. So with the same recipe, it can be at least for 5 different species of Lepidopteran insects. So that will make us a bit easier to conduct any rearing of the insects. Yeah.
Excellent. Now Davina talked about, I think she had a preferred, I think you said that the topical application, if I remember rightly, may have got that wrong, but the preferred sort of whether it was model to use, whether it was overlay, diet incorporation, dipping, I think you had potter spray there, topical application. Andy, what was your favorite though in all of those? I mean, for fall armyworm, what did you use most?
Fall armyworm, it can be what we did before. It can be using the diet overlay. With that, with the tray that we use, it can last for 7 days. But if we want to look at more than 7 days, and then we have to use different chamber, as probably Divina used, which is much bigger than what we had. So it's again depending on the time of the observation and then the space that we have and how how fast the larva is going to grow. That will determine everything that we need. But with the fall armyworm, I think topical application is going to be also very visible because the size of the larva is big enough. So using maybe the third instar larvae can be very workable for us. Even though probably, if I remember, we haven't done anything topical application for polar mealworm, right, Fallon? I think we have not done yet, but it could be done because
We done
we
it
did with it.
first. We
Oh,
done it
we did.
once. Yeah, but with
Oh, okay, we did.
third or fourth install.
Okay, yeah.
Excellent, Val.
So
Val, what did you find the hardest in doing the bioassay work? Because I know you did a lot helping out there. What was the hardest for you?
The hardest if yeah, for the topical application when you use the third or fourth install, that's hard because we have to provide a lot of larvae at the same time because after second install, they usually develop or grow with different stage. So because we have to use the larvae which have same weight, the same size.
Yeah.
That's the most difficult one. But when working with
Yeah.
neonates, that's full of happiness.
You sound like a nurse working with the neonates.
Oh,
That's
very well. When they're hatching, oh, so many of you.
Brilliant.
Very
I love it.
active. Very, very active. Yeah.
Excellent. I love it. Davina, you've got a question actually. Your little slide there on the US and Indonesian lure, and obviously the Indonesian lure. I mean, why am I not surprised? That works the best. What's the difference? Somebody
Yeah,
asks.
well, they're both aggregation pheromone, but the US lure is specifically for the Rhynchophorus ferruginous.
Yeah.
The Indonesian lure, which we got from one of our collaborators, they sent us because I was interested that it can trap the three species of Rhynchophorus as well as the rhinoceros beetle. And true enough, when we put it here in the Philippines, we caught Rhynchophorus and rhinoceros beetle on that. So I talked with our collaborator and I said that maybe we can partner in looking at the aggregation pheromone, because what my interest here, Andi, Tech, is the rhinoceros beetle, because we're getting rhinoceros beetle from the Indonesian lure, meaning that we have the same biotype of whatever rhinoceros. I brought this lure to Guam because Guam has only rhinoceros. They don't have red farm weevil. They don't have Rhynchophorus. They only have rhinoceros. When I put it there, it did not trap Rhynchophorus. Of course, they don't have Rhynchophorus. It did not trap also rhinoceros. So that is a very interesting.
The mystery.
Yeah, it's still on, and I think we can work on that. What is the true biotype in the Southeast Asia and the Guam and the Pacific? I don't know, something that I think we can think of as a collaborative research for us, for the group.
Excellent, Davina. No, I think it's a great collaborative project, but I'm going to throw it over to Tex soon because he loves those sorts of projects. That sounds just up to... Hey, do you think it's going to be as easy breeding, just on the bioassay thing first before we sort of open it a bit further, and we're almost at the end, but do you think it'll be just as easy to breed red palm weevil as it is for ball armyworm, or just as difficult depending on which?
Artificial diet, we come up with an artificial diet of red farm weevil just for the larva, because we would like to use larva incorporation, diet incorporation for the pesticide. So we need to come up with an artificial diet that will work at least for 3 stages, from the first instar to the third instar of larva of red farm weevil. And we made it. We were able to come up with a diet up to the I think 8 larval instars. So we're very confident that we're okay for bioassay up to the third instar of red farm wheat. But it's not easy. It's easy for us. We made it easier for the artificial diet that is working for us for fall armyworm than for red farm weevil.
Excellent. Thanks, Davina.
Yeah.
I'm going to throw it open to Tech to ask a few questions, or maybe a few points that maybe are important for people to consider when they're doing bioassays, and then we'll draw it to a close. Yes, Davina?
Listen, there's a question here.
Oh, yes.
Have you considered using ACV artificial diet from UPLB in mass reading? Yes, we were not able, we were asking for that. We were not able to get that from them. So we end up making our own diet, because I think during the time they're still testing
Okay, yeah.
the ACV diet. So they won't like to release the formula yet. So
Yep.
we cannot wait, no, because we have a research going or an experiment going. So we end up coming up with our own artificial diet.
Excellent. Thanks, Davina, and thanks for answering that question.
Yeah.
Brilliant. Tech.
Hey, thanks to all the speakers. Fantastic talk. A lot of work. Just because of time, I'm just going to ask maybe 2 questions and direct it to Davina.
Yes.
So the first one is about bioassays. Just wondering, are you trying to do bioassay for the Rhynchophorus? I didn't quite. I think I missed it.
Yes, we did. We already did the bioassay for the Rhynchophorus, because we were able to come up with a diet, artificial diet. And this was the master's student research, one of my master student, and I was not able to get the data right away, no. But the one that I showed is in the presentation, azadirachtin was the winning insecticide. That's what we use for the preliminary field trunk injection.
So just a follow-up to clarify on this question is, so which instar larvae are you using, and was it very difficult to get that going?
Which
For the bioassay?
stage?
Yeah,
Which
that's right.
stage? We did the first instar.
First instar, okay.
We did the first instar.
Okay.
Because I
Look,
think
fabulous work,
we
yeah.
were able to come up with a good low mortality with the first instar, because they eat too much. So
Yeah.
they really eat too much, so we need to come up with a good chunk of food. So we end up, I think we are confident that first instar larva will be able to give us a very good result on testing the different materials before we try it in the field.
Okay. And my second question, I guess more speculation than a question for you. So we all know why the fall armyworm is detected so widely and so rapidly across the world. Analysis suggested, multiple introductions and whatnot. I'm just wondering about, for the Rhynchophorus, how do you think that you have this pest population occurring in Philippines? What might be the mode of introduction, or how?
No,
Yeah.
we don't know yet, and I think that is one that I think we need to work on together. We can discuss this. I think Barbara is here. We have already a project that will soon start for the Philippines. But I already inquire from the HR if we can submit a proposal so that Southeast Asia is included in this, and it has to be through the STP card for us. So I don't know if we can have that, because we already have approved project for the Philippines. So Maybe we can talk about this with the Indonesian here to be the lead country, and we can help with the HR here in the Philippines on that. I can continue discussing with the HR group here in the Philippines on this, because it is very important. Like, my thought is, did you see the color morphs of the island, the separate island and the main island? And what really interests me is, why is there only one color morph in the little islands, and in the bigger islands, multiple column? And I think that is one that we need to look into and have an answer, because that will have an impact on the control strategy for this insect.